Hey, good afternoon, everyone. Welcome to our Friday training. I'm trying to do this weekly. This is the third week in a row that we're doing it weekly, and hopefully, whether you're in the Facebook group or the LinkedIn group, you're enjoying and benefiting from some of this knowledge. Again, I want to encourage everyone to invite your colleagues and friends to these groups and to ask questions, have discussions, post whatever information that relates to these topics that you're just not sure about, and be happy to answer them and interact and dialogue, even on top of these educational aspects. So, what I wanted to do today is I wanted to kind of dive back in a little bit here and talk a little bit more about connectivity and brain networks, which is something that we'll certainly talk a lot about in these groups because it's a basis of our understanding of the brain and of the way we do this neurofeedback treatment, this multivariate coherence neurofeedback treatment. So, um, this is certainly not a training or a course in understanding neuroanatomy and neural network, the connectome, because it's much more complicated than we can do here. Um, but I did want to just do a little introduction to understanding the idea of structural brain pathways, because as we're trying to understand pathway linkages that we might assess from the QEEG or other people or other techniques, we want to know what those networks are so we can assess if there are connections across them. So, for example, if in some sort of QEEG we find out that someone talks about a connection between T5 and FP1, well, there's no direct pathway that does that. So we want to, there are multiple pathways that'll get you there, but not a direct one. So we want to understand these pathways so as we see these assessment findings, and we can link them to the likelihood that a network exists to carry this information and that it's related to the individual symptoms. So um, Marco Catani has written probably the largest atlas of these networks, and they use MRI fusion tensor imaging images to display them both in the paper and in the textbook that they provide. So I would certainly recommend that for anyone who's interested in learning about such information. But basically, to summarize, there are some basic pathways we want to understand. So going from top left across the rows and down, we have the anterior commissure, which connects the frontal poles on both sides. We have the corpus callosum, which is the largest connection of pathways in the brain that connects the hemispheres from side to side, from front to back. It does not go all the way to the prefrontal cortex because the anterior commissure does that. We have the cortical spinal tract in the upper right-hand corner, which relays information all the way down to the spinal cord, cerebellum, through the basal ganglia, and up to the cortices related to motor information. And there is also one for sensory information that has a similar pathway. The cingulum in the middle left is what we usually talk about as the cingulate gyrus, which overlies the limbic system and by many is considered a part, sort of like the entrance into the limbic system. So that's a very important pathway. The fornix is very critical within the limbic system for memory functioning. The uncus fasciculus, um, uncinate fasciculus, connects the prefrontal cortex, inferior orbital frontal areas, and the anterior temporal lobes, and is very critical in emotional regulation and is often injured in traumatic brain injury. In the lower left, we've got the inferior frontal occipital fasciculus, which connects the occipital, temporal, and frontal regions. Next is the longitudinal inferior fasciculus, which connects occipital to temporal sites on the lower end of the brain. There is also a longitudinal superior fasciculus that goes above that, that's not pictured here. And then there is the arcuate fasciculus, especially in the left hemisphere, that connects speech and language centers of the brain. So there are many more, but it's important to understand these linkages and these known networks. This is another look at some of these pathways. So this is a great reference if you want to look at this one as well. So we've got imaging here of the inferior frontal occipital fasciculus, the uncinate fasciculus here in blue. These are all your association pathways. The cingulate in red, the superior longitudinal fasciculus, which we didn't see before, in red, the inferior longitudinal fasciculus in green, and then there's another portion of the cingulate that wraps around near the parahippocampal gyrus. There are also thalamic radiations. So the thalamus is the sensory relay station of the brain, and there are radiations from the thalamus up to all portions of the cortex, virtually every representation responsible for sensory input of any kind. So that's very critical. And then the projection fibers, we talked a little bit about the cortical spinal tract, which we see here in white, but we also have aspects of the forceps major and minor, the acoustic radiation from the thalamus, the medial lemniscus, and the middle cerebellar peduncle, which are projections across motor fiber areas as well. So again, this is not exhaustive, but if network pathology or network therapy is something you want to pursue, I would highly recommend that you learn a little bit about those known networks. And we know them from autopsy studies, but we also know them from these diffusion tensor imaging studies that we're learning more and more about. One of the ways we look at this complex information when it comes to neuroimaging is through a technique called graph theory analysis. It is a way of understanding network dynamics in a theory that sort of represents the environment of the brain. Graph theory was originally created based on social theory and civil engineering in terms of planning neighborhoods and networks and understanding how these things interact, and it's been applied to neuroscience in the last couple of decades. And we actually use it as part of our analyses now as well. So there's a couple of things that are very important to understand. You have what's called a node, which is a location, whatever that location may be in the brain. You have an edge or a path that connects nodes. You have high degree and low degree sections of this, and you have connectors and modules that exist together. A provincial hub is a hub is basically a region where nodes are all connected to each other, and a provincial hub is one where there's multiple connections that are larger than just a smaller connector hub or even a module, as you might see there. As you put all this together, it's very important to understand these graph theory elements are nodes, edges, hubs, modules, etc. And understanding that the connecting pathways that are viewed as edges, we can also refer to as a path. And what graph theory really does for us is it gives us a measurement of the efficiency of communication across paths. And that's what we really want to understand. Okay. So we've talked a lot about that. This is an idea of a provincial or a rich club network where you have hubs that are all connected in a network and all connected to each other. And the greatest rich club network that we know of is the default mode network in the brain. We'll probably talk about next time when we talk about functional brain networks. So this takes us to networks and path lengths. So again, very important to understand that these edges or paths form a certain form the communication going from node to node, hub to hub, module to module, etc. And we can measure, because EEG is so good at measuring timing, we can use the EEG to measure what's called path length or distance, which is the time it takes to get from one node or one module to another, what we call path length or path efficiency. So we talk about this idea of path length. Path length is a measurement of the speed with which it takes to get from node to node or module to module. The measurement of path length in graph theory metrics is an average of all paths, and then we can break it down into paths that are closer to each other, called radius or radii, and paths that are further apart from each other, which is what we refer to as diameter. So those are long-range connectivity and short-range connectivity concepts. Graph theory network analysis has been used to understand several different clinical conditions. This is a measurement of the default mode network that shows you the connection from frontal to parietal regions, which is what we understand that network to represent. And it's responsible for just us being at rest, us relaxing, being calmer, being able to use our attention and memory skills to their betterment, because the calmer we are, the better we can do that. And this looked at network analysis related to that and to its association with memory and anxiety. Anxiety is a major feature. When someone has dysfunction of the default mode network, they will almost always have anxiety. And that's very common in PTSD and developmental trauma as well. So this is a representation of a series of a study done in autism, where on the right-hand side you can see a typically developing brain, and this is a group of autistic individuals where what you see are modules that are very close to each other. So they're what we refer to as hyper-connected, and then others that are very apart from each other or hypo-connected. And this way of grouping the data makes this very easy to see and appreciate. And that's the typical type of pathology we see in individuals on the spectrum. Here's another just example of that, where you can see this large intersection here and this area over here where there's very little connection between areas. So that forms the basis for the core problem in autism, where there's local hyperconnectivity, but a lot of times, more importantly, there's longer distance hypoconnectivity. And we can measure this with graph theory metrics, which we'll get to when we look at our analyses and explain how those operate. This was a study we did back in 2017 when we looked at a group of controls compared to those who had developmental trauma in childhood, and showed, based on graph theory analyses, showed that the developmental trauma group had much greater dysfunction in connectivity, but especially in what we call the diameter, which is long-range connectivity over these periods of time, similar in some ways to what we saw in the autism example as well. So I hope this whets your appetite a little more about some concepts related to networks, and next week we will do a little more on that related to functional brain networks. Again, let's discuss. Please ask questions, make comments, like things, invite others, form your own conversations with people in the group. We really want this to be as interactive as possible and welcome as many people as possible to either our Facebook or LinkedIn group. All right, I hope everyone has a great week and talk to you soon. Bye-bye.